I'm Maria Costanzo. On behalf of the Knowledge Portal team and the Accelerating Medicines Partnership in Common Metabolic Diseases, I'd like to introduce you to a public resource intended to help researchers translate genetic associations into accessible knowledge, the Common Metabolic Diseases Knowledge Portal, or CMDKP. The central challenge of complex disease research is to discover the molecular mechanisms by which genetic variation impacts biological processes. Thousands of genetic associations have been discovered for common diseases and traits, but it's challenging to figure out exactly how this variation affects a disease. The goal of our project is to help researchers move more quickly along this path from association to function, in particular to accelerate the identification of the effector genes that act to impact disease risk. We can do this by integrating and analyzing multiple data types in order to make predictions about disease effectors and mechanisms. We work with experts in different disease areas to identify and aggregate the most authoritative datasets from cohorts around the globe. Associations are meta-analyzed using the overlap-aware METAL method to produce a bottom-line consensus view of associations for each disease and trait. Most of the variants that are associated with disease likely affect gene regulation, and the CMDKP includes multiple types of epigenomic data that can provide insights about the regulatory potential of genomic regions. We aggregate these data in collaboration with the Diabetes Epigenome Atlas. We then apply multiple bioinformatic methods to the data in order to predict connections between variants, genes, tissues, and pathways. Comprehensive results and predictions are displayed in the CMDKP. If you'd like a view focusing on a particular disease research community, you can navigate to any of 5 disease-specific portals. The CMDKP currently presents results from 3 methods that predict T2D effector genes. The curated list is the result of an heuristic that incorporates published genetic, regulatory, and mutant phenotype evidence. The other 2 methods, which are both more automated, use algorithms to combine evidence types and generate a probability score. The Genomic Region Miner allows you to explore the evidence about regulatory targets and tissues of action for a locus by stacking up genetic associations, epigenomic annotations, and credible sets across a region. This module is still in active development, and we welcome comments on it. The GAIT tool allows you to set multiple custom parameters and run real-time aggregation tests to ask whether a gene of interest is significantly associated with a disease or trait. We also offer pre-computed gene-level association scores generated using either common or rare variants. And the Gene Finder tool allows you to search MAGMA results to find genes associated with multiple phenotypes. These are just a few of the tools and displays available in the CMDKP. Please visit the site, let us know what you think using the contact form linked at the top of every page, and stay in touch with this effort on social media. Jason Flannick's talk at Endo 2021 will provide much more detail.